All right, we're going to go into, I moved the screen. We're going to go into sorry, I think that the waiting room has slowed down and starts talking, then it speeds up again. So we're going to go into nucleic acids today. And before I start into that, I want to point out that tomorrow we will have another PFG. Tomorrow will be like yesterday. You can log on and do them and ask questions if you have them, or you can just get it and log off and log back on if you have questions, or however that may work. I did want to point out to you folks that there is a common problem that people have with these naming things. Year after year, it's a common problem, and some of you are having it. And that problem is when people are pretty good at seeing things that are hanging off, shall we say, like a double bonded O, or if there's no H. But people frequently miss when there's two things, particularly an O stuck in the middle. I don't know if the curves of it look too much like a C and people don't pay attention. I don't know if that's it, but it's pretty common that when there is two things, 1, 2, that something gets missed. Even if it's an N sometimes, and you would think that the N with its lines and square and pointy corners would stand out, but when there's two things there, usually O's, but sometimes N's, people miss it. Also, a couple of you yesterday were missing the group 7 halogens. You noticed that there was something there, but you didn't know what it was. I can't remember what yesterday was. It might have been BR. But people were like, well, this is what's different, but then I don't know what that is. Well, if it's not nitrogen or oxygen or hydroxide, it's almost certainly a halogen, making it a halide. All right. Nucleic acids. Nucleic acids are complicated, so brace yourself. There is a lot of back and forth and overlap, and exceptions will, in this case it's this, but in that case it's that. So you're going to want to focus, you're going to want to be paying attention, you're going to want to spend some time reviewing it. Nucleic acids are, in my opinion, I think I said something about this, with proteins maybe, nucleic acids are, in my opinion, one of the best arguments there is for creation against evolution. The notion that these things could have come about, this complicated of a system could have come about by random chance, is just mind-boggling. And it's ingenious. Nucleic acids are essentially a 4-letter storage device that can copy itself and is capable of storing all of the information it takes to make any living thing anywhere. So when I say 4-letter 4-character storage device. Computers use a 2-character storage device. The characters would be 1 and 0, or I and O it's sometimes expressed as. That's why some on-off switches, I don't think it's as common as it used to be. Some on-off switches look like that because 1 and 0. Maybe this is a little longer, I don't know. 1 and 0. 1 and 0, on and off. The 2 switches, the 2 characters that computers use to store data. Well, nucleic acids use 4 characters. Basically, nucleic acids are like the, shall we say, the hard drive or flash drive of your cells, and they are the processing unit, the CPU of your cells, kind of all together in the same thing. So nucleic acids, subunit is nucleotides. Now, nucleotides are composed of 3 pieces. So in this case, the subunit has subunits. The subunit nucleic acids are made of nucleotides. Nucleotides are made of these 3 pieces. Like I said, this gets pretty involved, kind of a little bit on the confusing side. So we'll look at some pictures in a minute. The 3 parts are the phosphate group, the 3 subunits, if you want to look at it that way, are the phosphate group, the pentose sugar, you know, penthouse, like what a rich person lives in. No, wait, that's penthouse. Pent. Remember, you may remember from earlier, I told you that OSE meant sugar, and pent means 5. So pentose means a 5-carbon sugar. There are 2 kinds of 5-carbon sugars, ribose and deoxyribose. D means without. So there's ribose and ribose without an oxygen. I'll show you in a minute. Ribose is found in RNA, and that's what the R and RNA stands for, ribo. And deoxyribo, the D is the D in DNA. So deoxyribo, this is deoxyribonucleic acid, as opposed to ribonucleic acid. And then we have our nitrogenous bases. Now, nitrogenous may sound highfalutin. All nitrogenous means is they have nitrogen in them. There are 4, 5, 4, yeah, it's confusing. In DNA, there are 4 nucleic acids, nitrogenous bases, making up the different kinds of nucleotides. Adenine, thymine, cytosine, and guanine. In RNA, uracil replaces thymine in RNA only. Again, there's a lot here. I understand that. It is, it's going to take a while to sort it out and let it all soak in. That's par for the course. Don't be too freaked out right now. So you'll notice I've got these together as in pair, adenine and thymine, cytosine and guanine. The reason for that is that they always have to pair with each other. When they go together, that's how they have to go together. So let's try and make some sense of this. So you don't have to know this. Just trying to illustrate some things here, and don't get too Yep, my pen's working. Don't get too freaked out here. So in DNA, this is a strand of DNA, a double strand of DNA. DNA is double helix. So in DNA, we see that this is DNA is made of these pairs, these base pairs. All right, so if we look at this, there's the phosphate group, and there's the pentose sugar, and here is, as it says, pentose sugar, and this is one of the nitrogenous bases: nitrogen, 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 nitrogen. That's why they're called nitrogenous bases. Nah, that's probably not a good joke. If we were to, well, why is it's a base? Because it's basic, the opposite of acids. I mean, it's not strongly as a basic. Otherwise, it would just dissolve the cell, but it's a very, very weak base. It's just ever so slightly basic, which is the opposite of acidic. Now, this O right here is the one that goes away in the deoxyribonucleic acid. So we've got the phosphate, we've got the sugar, the pentose sugar, and we've got the nitrogenous base. Now, here is the same thing, all put together with 4 different bases. So thymine and adenine have to go together. Why do they have to go together? Because these hydrogen bonds, I don't quite get why there we go. You couldn't see it, I don't think, but my Zoom console wouldn't go away and I couldn't reach the controls for my pen. So you see these nitrogenous bonds here. I mean, hydrogen bonds. The hydrogen bonds are what hold adenine and thymine together. You will see there's 2 of them, and they line up. The chemicals that make them, the atoms that make them, line up. In the guanine and thymine, cytosine pair, we have 3 hydrogen bonds. All you have to remember is that they go together, guanine, cytosine, and they're held together by hydrogen bonds. I'm just showing you, if you care, why. Why do they have to go together? Because that's how they will form hydrogen bonds. If you tried to put these 2 together, the hydrogen bonds wouldn't line up right. If you tried to put 2 guanines together, it may be 3, but they don't line up right if you try to put guanine and guanine. So that's why they have to go together in that particular arrangement. You don't have to really remember the why. You just have to remember that adenine and thymine, doesn't matter which order you say it, adenine, thymine, thymine, adenine, have to go together when they pair up. Guanine and cytosine have to go together when they pair up, except that's with DNA. RNA, instead of thymine, it's uracil. All right. Oh, that's the same thing. So there you go, same thing. Now, here is the difference between DNA and RNA. DNA, which is what we've been looking at, has the 4 bases. Cytosine and guanine, which have to go together, and they're shown as blue and green. So you will notice that the blue and green always go together. Wherever you see a green, you see a blue, or vice versa. Although you can't see very much of the blue right there. There's a blue and a little smidgen of green showing through, blue and green, blue and green, blue and green. So cytosine and guanine always go together. Adenine and thymine always go together, which you'll see are shown as yellow and red. So yellow and red, yellow and red, yellow and red, two of them together. They have to pair up like that because of the hydrogen bonds. Now notice we don't really show the hydrogen bonds. They frequently just show that they have shapes, looks like a fish, frequently show that they have shapes that fit together, as opposed to here, they've got rounded instead of pointed. Now, DNA, RNA is usually single-stranded. See, we only have one strand running up here. We don't have two of them. Although sometimes it can temporarily pair with something else. So when RNA temporarily pairs with something else, cytosine and guanine still go together, just like over here. These are the same. But instead of adenine pairing with thymine, adenine pairs with uracil, M0. So instead of adenine thymine, we have adenine uracil. Now, adenine in DNA can pair with uracil in RNA. And that does happen sometimes. You don't have to understand all of this. I'm pretty sure that there's a lot more scribbles and material here than some of you are comfortable with. It's going to take a while to soak in. But that gives you a visual picture of what we've got going on with the bases pairing and the double strands and so on. Notice, can't remember which one we looked at here. We looked at, didn't tell you which nitrogenous base, all right? So notice cytosine, nitrogen, nitrogen, nitrogen. Guanine, nitrogen, 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 nitrogen. Adenine, nitrogen, 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 nitrogen. Thymine, nitrogen, nitrogen. Uracil, M0, nitrogen, nitrogen. So they all have nitrogen in, and they're basic, as in, not as opposed to, basic as in the opposite of acid. All right, so these have to go together, and you're going to need to remember that these have to go together. They have to pair with each other when they're paired. Whoa. And uracil pairs with adenine. Not with thymine. They replace each other. Now, already you've seen that we're starting to get into the subgroups. And the subgroups have differences and similarities. So that's why this starts to get annoying and somewhat difficult to keep track of. You know what? I don't want to break that in half. So I'm going to let you go right now, and we'll pick up on Friday with the deoxyribose, the subgroups, DNA and RNA. And remember, you've got a quiz to take. Remember, tomorrow we will have a Zoom meeting, but it will be a PFG help session. If you don't need the help, just get the PFG done. So have a good day.